Hello children, how are you? Fine? Hope you are enjoying my video classes. So, in continuation with the previous topic, a brief recapitulation, we have completed the principles of inheritance and variation, where we have discussed different aspects of gene, its transmission and related disease during abnormality. Now, we are starting another chapter which is very much related with this, that is molecular basis of inheritance. Molecular basis of inheritance. Under this topic, we shall be talking to you today DNA, the genetic material and its structure. Now, when we talk about DNA, apart from DNA, there is another genetic material called RNA. RNA. DNA or deoxyribonucleic acid, deoxyribonucleic acid and RNA, ribonucleic acid. Now, what is the basic difference and what is the most common genetic material? For all the higher organisms, the genetic material is DNA. So, DNA is the most abundant or common genetic material. Although in certain viruses we have RNA as genetic material. Say for example, as you know, the whole world is today victimized by the COVID-19. That is due to a virus, coronavirus, which is having a genetic material RNA. HIV virus having a genetic material RNA. Anyway, so let us make a difference of structure of DNA and RNA. What is the basic difference in DNA and in RNA? So, this is the DNA and this is the RNA. In the case of a DNA, we have a sugar molecule. In the case of RNA also, we have a sugar molecule. But you have to be very, very careful about the structure of the sugar molecule. This sugar molecule of DNA having see the structure, carbon number 1, carbon number 2, carbon number 3, carbon number 4, this is 4 and this is 5th. So, it is a pentose sugar, name is deoxyribose. And here in case of RNA, it is also a pentose sugar, but the type of sugar is a ribose sugar. What is the difference? See the structural difference. Carbon number 1, carbon number 2, 3, carbon number 4 and 5. So here see the difference. So this is deoxyribose or DNA having a pentose sugar in the second position of carbon. This is the second position of carbon. You see there is a hydrogen but not hydroxyl group. In the case of ribose sugar, which is also a pentose sugar means sugar having 5 carbon. Is it clear? Now, both are pentose sugar. Deoxyribose is also a 5 carbon sugar. Ribose is also a 5 carbon sugar. But the basic difference is, in the case of deoxyribose sugar, there is hydrogen at the second carbon position. Instead of that, in ribose sugar, there is hydroxyl group. So additional hydroxyl group at carbon position 2 is present in the case of RNA, which makes RNA more reactive. So if you compare about DNA and RNA, RNA is more reactive and less stable, whereas DNA is less reactive and more reactable. When we shall talk about which is the best genetic material, that time this quality again we shall explain. Now it is not the nucleotide or nucleoside. It is a phosphate sugar. Now it is a pentose sugar. Now this pentose sugar, when it will attach with a nitrogen base, when it will attach with a nitrogen base, then it will form nucleotide and nucleoside. Now it is nucleoside. Nucleoside. This is also nucleoside. So this is deoxyribose nucleoside. This is ribose nucleoside. So if I write nucleoside, is that clear to you? Deoxyribonucleoside, ribonucleoside. Now, what are those nitrogen bases? Nitrogen base, so let me write here nitrogen base. These are of two types, purine and pyrimidine. Purine again are of two types. What are those two types? Adenine and guanine, whereas pyrimidine are of cytosine, thymine, amine, little less space, cytosine, thymine, or in place of thymine it can be uracil. So remember again one more difference. In the case of DNA, the nitrogen base are, in the case of DNA, the nitrogen base, N base, are adenine, guanine, cytosine and thymine. That is T. Symbolically I am representing. So in the case of DNA, the nitrogen bases are adenine, guanine, cytosine and thymine. Whereas in case of RNA, DNA, in case of RNA, the nitrogen base are adenine, guanine, cytosine and uracil. So remember, where DNA contains thymine, RNA contains uracil in place of thymine. Is it very clear? Now, let us go a little deep. In that sense, let us have a structural composition of purine and pyrimidine. So, if I rub this part to make little accommodation, purine, that is adenine and guanine, are a 9-member ring. Are a 9-member ring. That is carbon number 1, 2, 3, 4, 5, 6, 7, 8 and 9. This is about adenine. Where in adenine, other carbon I am not talking about. Here there is NH2. 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 Double bond, of course. And in case of adenine, A. In the case of guanine, again here, it is a 9-member ring, a benzene ring 6-member and a pyrenoid 5-member. But then what is the basic difference? Here, in the case of guanine, it will be oxygen. Oxygen. The remaining thing I am not giving because in class 11th standard you have already studied this structure. Now, what about cytosine, thymine and uracil? In cytosine, we have a 6-member ring like this. Here it is NH2. Cytosine. In the case cytosine, in the case of thymine, it is again 6-membered ring, but here is oxygen. Remaining thing I am not drawing. So carbon number 1, 2, 3, 4, 5 and 6. In the case of thymine, again here 1, 2, 3 carbon positions, 4, 5 and 6. And uracil, same way, again 6-member ring. Here also there is oxygen. 1, 2, 3, 4, 5, 6. So, I have given you the structure of nitrogen bases. Now, when the nitrogen base, let me rub this part and go a little more elaborate one. So, when the nitrogen base, suppose this adenine nitrogen base combines, how it will combine? How it will combine? This is a 6-member ring. 5-member. So, this is NH2, carbon number 1, carbon number 2, 3, 4, 5, 6, 7, 8 and 9. Add this 9th carbon, the first the first carbon of pentose sugar deoxyribose will combine with the 9th carbon of adenine, thereby forming a deoxyribonucleoside. But when it will combine with a phosphate group at its 5th position, suppose phosphate group. So this is the phosphate group. There is an oxygen here. Phosphate group. Phosphorus is having 5 valence electron, 1, 2, 3, 4, 5. 1, 2, 3, 4 and 5. This one. With this 1 electron. So, now the total component became a nucleotide. Nucleotide. So this is one nucleotide. Then what is DNA? DNA is nothing but a polynucleotide chain. So let me write the definition of DNA. DNA is a polynucleotide chain. Means many such nucleotide will together combine one to other, thereby forming a polynucleotide chain. That is what we call a single strand DNA. Now, in that nucleotide, 3 things are there: nitrogen base, phosphate sugar, your pentose sugar, and phosphate group. The phosphate group and pentose sugar forms the backbone of DNA, whereas this nitrogen base remain inside the 2 antiparallel strand, in between the 2 antiparallel strand of a DNA, forming a ladder of a staircase, So, when at 5 position one phosphate group is attached, this is called 5' end of nucleotide. Here the next nucleotide, when it will attach, the phosphate group will attach here at the 3' end. So another phosphate group will attach with this. Another phosphate group will attach. Like that way, another nucleotide will attach to form a dinucleotide. Likewise, 3 nucleotide, trinucleotide, tetra, penta, hexa, hepta, octa. Like that way, numerous nucleotide will combine to form a DNA strand. DNA having 2 strands, 2 such polynucleotide running antiparallelly in a right hand helix. Just look at my hand. This is one polynucleotide, left hand, this is another. They are running like this, right hand helix. So if I put my duster here and chalk here, see here the 2 hands, one polynucleotide, another polynucleotide, how they are combining in the form of a right hand helix. This, like this. This is the structure. This structure was discovered by Watson and Crick, the famous scientist who has been awarded with Nobel Prize because of their excellent discovery of structure of DNA, double helix structure. Now, if we draw a double helix structure of DNA, how does it look? Let me rub this cushion to make little space. This is one strand of DNA. One strand of DNA. And This is another strand of DNA. Suppose this is the 5' end of this polynucleotide chain. This is the 3' end. 3' end. So the other chain will be just anti-parallel, like this, running like this, like this. So this will be the 3' end, this is the 5' end. That is what you call anti-parallel. And this backbone is made up of phosphate and pentose sugar deoxyribose. So this backbone is this, made up of phosphate and pentose sugar. Where does the nitrogen base reside? Inside, in the form of a ladder. Suppose this is adenine. So there will be anti-parallel complementary base pairing. Adenine will combine with thymine. That is called AT pairing. Guanine will combine with cytosine. While adenine will combine with thymine, there will be a double bond. Double bond. This is the hydrogen bonding. And while guanine combines with cytosine, there will be triple bond. Likewise, adenine to thymine, guanine to by triple bond to cytosine, and like that way it will continue. So a little more adenine AT. So this is what you call AT-CG bonding. CG, where A combines by double bond with T, C combined by triple bond with G. AT, C, G. In that way it will continue. Now, how many such nucleotide might reside in DNA? Or what will be the length of a DNA? If we want to calculate the length of a DNA, see the number of nucleotide or the length we calculate in the form of how many base pairs are there. Remember, in between this, the distance between this to this, what we call a pitch. Within this pitch, the distance is, what is the distance? In between two base pair, the distance is 0.34 nm, nanometer, and 1 nm, nanometer is equal to 10 to the power minus 9 meter. So within this pitch, how many bases are there? 10 bases, 10 nitrogen bases, 10 bases. Okay? So what will be the distance? You can easily calculate from this point to this point. That is 0.34 into 10 is equal to 3.4 nanometer. In case of, say, virus 5174, this is one virus. The total number of base pair is roughly 5386 number BP. In case of E. coli, Escherichia coli, a very common bacteria, E. coli is having nearly 4.6 into 10 to the power 6 base pair. What about human being? If we take a single DNA strand of human being, suppose 5' end, 3' end. In this single base pair, how many nitrogen bases are there? That is 3.3 into 10 to the power 9 BP. 3.3 into 10 to the power 9 base pair. So, if we consider both these strands, that means two strands are running anti-parallelly. That means number of base pair will be 2 into 3.3 into 10 to the power 9 is equal to 6.6 into 10 to the power 9 BP. This much base pair human DNA is having in diploid cell. In haploid cell, this is the number of base pair. So, what should be the length of DNA? We can easily calculate applying the mathematical sense. The length of a DNA, we are having 6.6 into 10 to the power 9 base pair, and the difference between two base pair is 0.34 into, if you convert it into meter, 10 to the power minus 9, this much meter. This gives you roughly around 2.2 meter. This is the length of a DNA. So human DNA is having this much length. And once we know the total structure of DNA, it will be very easy for you to understand the remaining concept of this chapter, which will help you to understand why DNA is considered as a better genetic material. Although RNA is also a genetic material, but DNA is considered as a better genetic material for transformation or transmission of the character from one generation to other generation. With this, I will stop today. For next part, we shall take up tomorrow. Thank you.